This is the title of my talk today. So I'm emeritus at Aix-Marseille University, and I'm also affiliated with the Quantum Biology Lab at Howard University. And what I'm talking about is the outcome of a long collaboration, interdisciplinary collaborations between theoretical physicists, my group at Center for Theoretical Physics in Marseille, molecular biologists at the Center for Immunology on my campus, biochemists at INSERM, which is the National Institute for Medical Research in France, and experimental physicists in Marseille, in Montpellier University, and at the University of Rome, La Sapienza. Okay. Now, talking with my colleagues and friends, biologists, I was confronted with a stunning situation of, say, molecular biology. In cells, as you well know, there is a huge number of biochemical reactions taking place continuously, and we know about 30,000 such reactions, if I'm right. And okay, this is a cartoon, which is a very simplified picture of what happens. But if you go on the cake depository in Japan, for example, this is an example of a small network of biochemical reactions inside the cell. This is a metabolic pathway. Each node corresponds, if you click on each node, you know what kind of molecules this node corresponds to. And each link tells how these molecules react. And so this is a small subset of this stunning complex network of biomolecular reactions. And so, as a physicist, one wonders how the hell is it possible that the cognate partners meet each other, being themselves at the right place, at the right moment, and in a good sequence. So how can they find each other inside the cell? The current explanation is that since we have a lot of water and each molecule, each large molecule is hit continuously and randomly by the water molecules, all these biochemical actor moves randomly inside the cell. So each biomolecule makes a Brownian diffusion inside the cell. And so the idea, the common wisdom is that sooner or later they will meet. The cognate partner of a biochemical reaction will meet. But what we call a back-on-the-envelope estimate tells us that this cannot work, actually, because we have a low concentration of interacting molecules, and so the waiting time would be very, very, very large with respect to what actually happens inside cells. And so we can wonder whether other kind of mechanism, of physical mechanism, could be in action. And since biomolecules are charged and have electric dipole moments, of course, the first idea is to think of electrostatic interactions. So we have charge interactions, charge dipole, and dipole. The problem, we have two problems. The device screening, inside cells, we have a lot of ions, and so the device screening makes the electrostatic interactions very short range, say less than 10 angstroms. Moreover, the water permittivity, the dielectric constant, is very large. It's about 80, and this weakens further the electrostatic interactions, and so the electrostatic interactions are ruled out. But again, since we have charged molecules with usually dipole moments, we can wonder what happens if we have oscillating electric fields instead of static electric fields. In other words, if charges and dipoles vibrate sufficiently fast, we have two things happening. The Debye screening, let's say, is weakened, and there are showing that actually at high frequency the water becomes and if water and ions becomes a transparent medium, and the electric dipole and the water permittivity at sufficiently high frequency drops down from 80 to a few units. In other words, if we think of oscillating electric fields, this can go very, very far away. In other words, the medium, even with the presence of ions, it becomes transparent, and say the water permittivity drops to small values. And so some years ago, we have investigated how this could work. And there is a paper which is cited here, where we investigated how the oscillating dipoles can interact through electromagnetic fields, actually, and what happens when two dipoles resonate at the same frequency or are off resonance. And we did this because many years ago, actually at the end of the 60s of the last century, Herbert Fröhlich, a distinguished physicist, theoretical physicist, put forward two main ideas. The first one was the idea that a macromolecule, which usually at equilibrium with at thermal equilibrium with its environment, wobbles, moves incoherently. All the atoms move randomly and independently, so to say, one of the other. If fed with an external source of energy under given conditions, could undergo a phenomenon which we can call a phonon condensation. In other words, some collective coherent vibration of the entire molecule could take place, and the expected range of these vibrations is around sub-terahertz and terahertz frequency range. And the idea that Fröhlich put forward, and we have revisited the original proposals by Herbert Fröhlich. There are some technical details I don't want to enter. The idea is that if the two dipoles, so associated with two macromolecules, for example, vibrate at different frequencies, so the system is off resonance, the electrodynamic interactions among these non-resonating dipoles falls off with the sixth power of the distance. Whereas at resonance, this becomes a long range. So the first one is a short range with the same power law of van der Waals interactions, but it is not van der Waals. This is with real exchange of photons, so to speak, instead of exchange of virtual photons. Whereas at resonance, this becomes a long-range interaction, and it falls off with the third power of intermolecular distance. And so this could be interesting because, of course, at resonance means selectivity. Because, in other words, if we have two molecules resonating, vibrating at the same frequency, instead of having a short range electrodynamic interaction, they could have a long range electrodynamic interaction. Now, the problem is that, the scheme is that Fröhlich condensation, in other words, by feeding energy on a macromolecule under suitable conditions, we could this molecule could undergo a Bose-like condensation. The Bose-Einstein condensation takes place by lowering the temperature for an isolated system, whereas a macromolecule in an environment, it is a dissipative system. In other words, it dissipates the energy which is injected by a non-thermal source. And if this happens, and there is a threshold for the rate of energy injection, if this happens, if this condensation takes place, we have, in principle, an associated giant dipole oscillation, which, as I told you, hasn't the device screening and can, at resonance, can act on a companion by switching on an interaction, an attractive resonance interaction. So this is a summary of what I just told you. The problem is that so the Fröhlich suggestions have been marginalized and forgotten for several reasons. First of all, the original work by Herbert Fröhlich was in a quantum mechanical framework. And we have the standard objections that biomolecules, Fröhlich, by the way, Fröhlich proposed to put forward these ideas to explain the high efficiency of enzymatic reactions. This was his motivating theme and topic. But then, since the biological environment inside cells is wet, warm, and noisy, and this means that quantum mechanical treatment is considered unsuitable. The Debray wavelength, the thermal wavelength, is so small that people said, "Okay, this is untenable." And of course, there is another practical problem. That is, even if these electrodynamic interactions between resonating objects, resonating macromolecules takes place, how can you detect them experimentally? Okay, so for this reason, these proposals have been forgotten and marginalized for a long time. But then, with some clever student of mine, we have proceeded to dequantize the original model put forward by Herbert Fröhlich. And instead of the theoretical chain of actions that you have on the left, you have a quantum Hamiltonian, you write the Liouville-Farnovian equation for the evolution of the density matrix. Then you change, then you make some averages, and at the end you get some rate equations telling you how during time each vibrational mode is populated. And then under some special conditions, when the energy input rate exceeds some threshold, you see that the energy fed to a macromolecule schematized with a collection of oscillators with nonlinear interactions and so on. You see that when the energy input rate exceeds some threshold, the energy is channeled to the lowest mode. This is Bose-Einstein-like condensation for an open system. And we have rephrased everything in a classical framework. There are standard techniques to do the same logical pathway in a classical framework. And okay, this is just for fun. The Austin Hamiltonian model in second quantization, which is behind the Fröhlich model. In other words, this is the starting point to prove what Fröhlich derived, so this phonon condensation mechanism. And again, this is for fun. I don't want to give you all the details. These are published. If you are interested, I can send you the papers. But the message of this slide is that we can get a set of differential equations describing the amplitude of the vibrational modes of a macromolecule subject to an energy input, this small s sub omega I. So we have an input, an injection of energy from an external source. We have a dissipation term, and then we have a very complicated set of nonlinear interactions among these vibrating modes. And so we can study what happens in this classical framework. This set of differential equations shows the same mechanism which was predicted by Fröhlich. In other words, for example, these are figures out of a paper that we published many years ago. We start, these panels are shown at different input energy input rates. And we start with an almost equipartition spectrum. These are the amplitudes of different modes. In abscissas, you have the frequency of different modes, different frequencies. And what happens when you increase the energy input rate? You see this green spectrum, you deplete the high frequency modes. And if you go on, it is the fundamental mode of lowest frequency which gets more populated, and all the others are depleted. And this panel shows a threshold phenomenon. Of course, it becomes sharper and sharper as you increase the number of modes. And here, for example, as a function of the energy input rate, this parameter S, you see that only one mode takes all the energy, and the others go down, lose energy, and go to very small values. In other words, also in a classical framework, a macromolecule modeled as a collection of vibrational modes that are nonlinearly coupled and considered as an open system. In other words, you have many mechanisms of dissipation of the energy that you inject into this collection of nonlinearly coupled vibrational modes. and you have an energy input. So it is like a quote-unquote laser. It is a standard of open systems. It undergoes a phase transition, a non-equilibrium phase transition corresponding to this phonon condensation. And so, having overcome the most important objection to the original Fröhlich proposal, that in a quantum mechanical framework you cannot, a quantum mechanical framework is not suitable for an object living inside a warm, wet, noisy environment. We tackled the other problem. If these interactions, in other words, once we have the phonon condensation and the activation of a vibrating giant dipole, this vibrating giant dipole could resonate with another macromolecule vibrating at the same frequency, and the prediction is that in principle we can have an attractive long-range electrodynamic interaction. In other words, they can see each other at large distances, but it is what is unknown and what is impossible to predict a priori is the strength of this interaction, of this electrodynamic interaction, even if these are actually present. In other words, they could be so weak that we cannot actually measure them. And another problem is that if they are present, how can we measure these interactions? And so what we did, we had in mind, of course, this came by discussing with our colleagues, the molecular biologists. We studied what happens if we consider a randomly driven movement. You see here these equations. These are Langevin equations. Forget this intermediate term in the right-hand side. And if we have just the noise, just the thermal random noise, this Langevin equation describes a diffusive Brownian motion of a given object, say a macromolecule. But now, if we have a collection of n molecules that interact through some given force, say, for example, these hypothetical electrodynamic interactions. So we have this drift term entering the equations of motion of n molecules that now, instead of being independent one of the other, now they are interacting. What happens to the diffusion coefficient, for example? The diffusion coefficient is computed by means of a standard methodology once you integrate it numerically, because you cannot do anything else, the Langevin equations. So the idea was, okay, let us see if we can, if these forces are sufficiently strong that we can experimentally detect them. Let us see if, by studying how a collection of n molecules diffuse in presence of these interactions. And of course, how can we study experimentally the diffusion properties of some molecular switch? For example, with fluorescence correlation spectroscopy, my colleagues, the molecular biologists, are expert of this experimental methodology. I assume that you know how things work, but you have a confocal microscope which illuminates a very small volume, typically of a few femtoliters, and when a fluorescent object passes through the observational volume, then the fluorescence radiation is collected back by the confocal microscope, and you observe a glitch produced by this molecule. Of course, when you have many molecules passing through the observational volume and randomly, of course, what you get here, the panel on the bottom left gives you a typical example of what you get. This is the fluorescence measured by the apparatus. And there is a standard technique computing an autocorrelation function of this signal that gives us the average transit time of a molecule crossing the observational volume. And this allows to measure the diffusion coefficient, the average, of course, diffusion coefficient. Then, in order to validate the fluorescence correlation spectroscopy methodology to test whether some molecules actually interact through long-range interactions, we studied, first of all, how oppositely charged molecules, we used the lysozyme and Alexa 488 in solution. And Alexa is a fluorochrome, it's something that emits light when it crosses the observational volume, and it is excited by the laser and remits fluorescence radiation. And so we can, having a collection of oppositely charged objects with a built-in long-range interaction, in this case, an electrostatic interaction. We studied how the diffusion coefficient normalized to the Brownian value varies when we change the concentration of these molecules in pure water or with an increase in concentration of salt, which makes it like screening. And when we have pure water, so we do not screen the electrostatic interactions, when the concentration of these molecules is larger, the electrostatic interaction wins against the thermal noise, and we observe a decrease of the diffusion coefficients due to the fact that the opposite charged molecules attract each other and are stuck one to the other, and so diffuse is lower. And in presence of salt, of course, we see the effect of the device screening. And on the other side, we made numerical simulations, which are in perfect agreement with the experimental outcomes shown in the left panel. And so this was an intermediate step to validate the experimental procedure that uses fluorescence correlation spectroscopy to check, to probe the existence of long-range interactions. Of course, these are built in. Of course, these are not what we are after, actually. So the electrodynamic interactions. So this was just to validate the experimental protocol. And then we made two major steps forward in this program. In other words, and we published several years ago the experimental observation of the phonon condensation in a protein. The protein that we studied was a BSA, that provides sermalbumin, and in this paper we have a theoretical part, which is the dequantization of the Fröhlich model, and so a theoretical part, and the experimental part, where the results of two different experiments performed in Montpellier and in Rome gave us the same result, and I will show you what this result is. And then another step forward was actually the experimental detection of the activation of these long-range electrodynamic forces. So we had two different setups for the first step in Montpellier and in Rome. These are so in Montpellier they use a nanowire and a waveguide to perform terahertz or sub-terahertz spectroscopy on solutions. And in Rome, they use a somewhat different. They use a bow-tie antenna on a semiconductor, and the detection of terahertz radiation is made by a two-dimensional layer of electrons. Okay, well, if you want, we can enter some details. So, as I told you a moment ago, the biomolecule that we considered was the BSA protein, and we attached between 5 and 6 fluorochromes, the Alexa 488, to each protein, and an argon laser is tuned to the absorption frequency of the fluorochromes. And the fluorochromes re-emit the absorbed blue radiation in the green, and the energy difference between each photon absorbed and the emitted one is about 0.2 electron volt per photon. Okay, so in other words, in so doing, of course, the BSA is in solution in water, and shining this laser on this solution excites the Alexa fluorochromes on each molecule. We create hot points on each molecule, and then there is a down conversion of this excess of energy of 0.2 electron volt per photon from the electronic part, from the electrons, the fluorochromes, we excite the electrons of the fluorochromes, but there is a down conversion of the absorbed energy towards the phonons. And we have studied also with numerical simulations how this happens and how it works. So these hot points give energy to the phonons, and what happens here is again how the two experimental setups in Montpellier and Rome were organized. So we have a watery solution of BSA proteins. We have the laser light, which makes the excitation of all these molecules, and the near field nano wire impinging in the solution, and a terahertz source, which is just a reading of what happens to the molecules, and the terahertz source makes a sweep in frequency. The setup, the experimental setup in Rome is somewhat similar, but different in the sense that the detector is a two-dimensional layer of electrons on a semiconductor. It is a FET, actually, substrate, and with about an antenna. So it is a different detector of terahertz frequency radiation. And okay, this is a cartoon. Now, the outcome in Rome and in Montpellier, the outcome in Rome is the red, are the red points here. In Montpellier, the black points show a sharp absorption feature at 314 gigahertz. And experimentally, both experiments observed the existence of a threshold. In other words, the input power rate has to exceed some threshold in order to observe the appearance of this absorption feature, and the amplitude of this absorption feature has a saturation when the optical power is increased. These features are qualitatively recovered by our theoretical model. In other words, the existence of a threshold. Of course, we have a small number of vibrational modes. When increasing the number of modes, this gets sharper and sharper. We have the saturation phenomenon. So qualitatively, we have an agreement. But the most interesting thing is that if we consider the formula giving the frequency of a spheroidal mode of an elastic sphere, this is found in the literature, and if we enter the Young modulus, the hydrodynamic radius, and the density, all these parameters for the BSA molecule are known experimentally. We put everything in this small formula, we get 308 gigahertz, so a few percent of difference. And this tells us what? This tells us that actually what we are observing is a collective motion. It is a global vibration of the molecule. So it is what we have called from condensation. This is a scheme. I can skip it because I have to run. We also considered, and it has been the right choice to detect the long-range electrodynamic interactions. We also considered the R-Phycoerythrin, which is a light harvesting protein which is present in bacteria, algae, and plants. It is a 240 kilodalton, whereas the BSA is, if I remember correctly, 66,000 66 kilodalton. This is 240 kilodalton. And so being larger, the collective vibration which has been detected for the Phycoerythrin takes place at the lower frequency of 71 gigahertz. And it has been a good choice because this protein, since the nature selected it as a light harvesting protein, has more than 30 chromophores which are naturally expressed together with the protein. And this is the spectrum, the terahertz spectrum, where we observed two frequencies at 71 gigahertz and 96. And of course, we cannot apply directly this formula, which gives us the frequencies of the different modes of vibration of macroscopic oscillations of a torus, of an elastic torus in this case. But if we take n equal to 0 and n equal to 1, the ratio of the frequencies, so these are stretching modes of an elastic torus, is the square root of 2. And what we observed experimentally is 1.35, which is largely satisfactory. And okay, then by increasing the laser power of acted on a solution of Phycoerythrin molecules, we observed the appearance of these absorption features. This means that these molecules vibrate at these frequencies, okay? And the experimental outcome, right upper panel, when we change the concentration of red Phycoerythrin in watery solution, the diffusion coefficient normalized to the Brownian value at low concentration gives essentially Brownian diffusion. When all of the sudden, when we increase the concentration, and so the average intermolecular distance decreases, all of the sudden we observe a drop of the diffusion coefficient. And this takes place at different concentration values according to the laser power. If we decrease the laser power, this happens at a higher concentration. What does this mean? By studying theoretically, semi-analytically, and numerically what happens to a collection of n interacting molecules with the same parameters of the molecule considered here, we observe a phase transition. In other words, this is reversed because what we change in our numerical simulations and in our semi-analytical model is the average intermolecular distance. And when the average intermolecular distance is large, we have Brownian diffusion of these objects. And all of the sudden, when the average distance is shortened, we observe a clustering phase transition. Here in the bottom right panel, we observe the outcome of Monte Carlo simulations, and all of a sudden, instead of having these dispersed molecules, we have the formation of clusters. We have a clustering phase transition. And experimentally, when here in the abscissas, so to speak, we have time, the observation time, and in the ordinates, increasing molecular concentration. So the baseline is without excitation, without laser excitation. But when we switch on the laser and we increase the molecular concentration, we observe after some time the formation of clusters. We switch off the laser, the excitant, and the clusters disappear. We switch again it on, and we have again the formation of cluster. This is a weaker phenomenon for a smaller molecular concentration. Okay, so we have movies of the appearance of these clusters when the laser power input is sufficiently stronger. When the laser power input is not strong enough, we do not see any cluster. So summarizing this part, I've been too slow because I have a second part. What we have found the first experimental evidence of this of the possibility of having phonon condensation. In other words, of exciting these collective intramolecular oscillations. And we have also a first proof of concept of the possibility of activating through this phonon condensation mechanism, the possibility of activating electrodynamic intermolecular interactions of sizable strength, because a priori, as I told you, it wasn't clear whether even if these interactions could be excited, could be activated, the question was, but can these be strong enough to be experimentally detectable? The answer is yes. Of course, there is a long way to go because we have considered identical molecules. In other words, the co-resonance, which activates long interaction and long distance interactions, have been observed with identical molecules. So the co-resonance was built in. And let me go back for a moment. What is interesting is that this interaction goes as far as, in this case, 1000 angstroms. And all the chemical interactions, van der Waals interactions, the bi-shielded electrostatic interactions are very, very short range interactions. Few angstroms, 4, 5 angstroms, 6 angstroms, van der Waals or Ramacher interactions, the screened electrostatic interactions less than 10 angstroms. And so these are very long distance interactions, 1000 angstroms, in comparison with all the other interactions. And of course, the million dollar question now is what happens if we have biomolecules of different kinds? In other words, the cognate partners of biochemical reactions. For example, this is interesting for ligand receptor interactions. So these are of the kind protein-protein interactions. And what happens when we have DNA-protein interactions or RNA-protein interactions? So this is of great importance for example, for the machinery of the transcription machinery. And these are open problems now. And another major open problem is, of course, inside cells we do not have lasers, and not all the proteins, not all the biomolecules have chromophores, as is the case of the R-Phycoerythrin. And so the question is, what can be the biological mechanism capable of activating these phonon condensations, and so activating these electrodynamic interactions? Of course, the first candidate and the most natural candidate is ATP hydrolysis. But, for example, also an asymmetric, an anisotropic momentum transfer due to the collisions of ions. The biomolecules are not uniformly charged, and so we can think that the collisions with environmental ions could make a momentum transfer anisotropic, and so this could make deformations, elastic deformation, and in the end this could produce these collective vibrations. So the way towards biology is still somewhat long too. And so wait, what happens? Okay, and so I have only a few minutes left, I guess. Okay, so we have started an exploratory theoretical investigation of other possible mechanisms, and we have studied the possibility of activating long-range, long-distance electrodynamic interactions between, for example, a protein and DNA. We have studied a restriction enzyme EcoR1 and an oligonucleotide. And in few words, we have a sequence of nucleotides of a given oligonucleotide with a subsequence which is a recognition site where the enzyme attaches and cleaves, cuts the DNA. And we have borrowed a model describing the interaction of electrons moving along a lattice. And we introduced in this model a novelty. So the upper line describes the motion, the hopping of an electron along a lattice, and the subscript n labels the site along the lattice. And then we have a phonon part, so there an elastic description of an elastic substrate where an electron moves, and then we have the interaction. And this can describe the motion of an electron along the backbone of a protein or along DNA DNA molecule. Okay, what we introduced, you see this Jn sub n and this chi sub n. The J sub n is the probability of jumping of an electron from one side to another, backward or forward. And chi n is the strength of the coupling of the electron with the elastic substrate, which can be a substrate of nucleotides or amino acids, according to the description of a DNA or a protein. And instead of taking this Jn and chi n constant, as in the case of the Davidoff model, we inserted a site dependence of these coupling constants. And so that we can model a given sequence of nucleotides or a given sequence of amino acids, so a given protein. And in the literature, we have the values of the interaction energy of an electron moving along a sequence of amino acids or nucleotides. When the electron finds an amino acid, here we have the list of amino acids, the interaction energy has a different value. And when it sees below him a nucleotide, also we have different interaction energies. And this allows us to insert the fingerprint of a given molecule in the model. And so I will make a long story short because my time is already gone. And if you want, we can discuss whenever you want all the details of the model. But there is a technique which is called the TDFP, time-dependent variational principle, which allows to write a set of differential equations which are formally classical and whose time averages coincide with the quantum averages performed on the quantum model. And okay, this is a long story, but what is interesting is that we can associate a formally classical dynamical system where the Cn are related with the electron, with the amplitude of the wave function of the electron along a chain of amino acids, so along a backbone of a protein or along a sequence of nucleotides. And the beta n are the amplitudes of the oscillations of the nucleotides or amino acids when the electron moves and excites this substrate. And so, okay, this is a long story. So we have currents. We have currents. The electrons move back and forth along a DNA fragment or along the backbone of a protein. So we have currents, and since the system, the lattice, the underlying lattice is not homogeneous, the frequency spectrum of each current is rich. And so what we can do, we can measure the cross spectrum, the cross frequency Fourier spectrum of the currents, say, taking place along the oligonucleotide, so along this fragment of DNA, and along the enzyme. the enzyme which recognizes a subsequence of this oligonucleotide. And the impressive outcome is that we find a co-resonance, a sharp co-resonance. When we randomize the cleaving site, so the subsequence which is recognized by the enzyme, this co-resonance disappears. So this is very interesting. This is a first step toward understanding how can we think of the activation of electrodynamic interactions between the cognate partners of a biochemical reaction. Of course, when we have a current, the D'Alembert equation tells us how an electromagnetic field is generated. This is the vector potential. And of course, J2 couples with A1, so with the electromagnetic field generated by the molecule 1, and vice versa. The current, say, of DNA couples with the field produced by the enzyme. And this interaction energy, when we make a time average, if J and A have the same frequency, these are the D'Alembert equation is a linear equation, so it inherits the same spectral properties of the current. And so when we have a co-resonance, the average is large. If the frequencies are not the same, if there is a frequency mismatch, this interaction energy in time averages to a small value or even to zero. Okay. Okay. This slide was to call the attention on the fact that water plays a major role. It is not, so to speak, just a witness of what happens, but the dipolar waves can be excited, and so water can play a major role. There is a nice paper which is referenced here down, calling attention on this fact. And a final slide to tell you that if we don't normalize the measured diffusion coefficient of the BSA molecule to the Brownian value, we observe, by increasing the laser power, we observe an increasing coefficient, diffusion coefficient. The Brownian value of a passive diffusion is about 50 micrometer squared second minus 1, the Brownian. Whereas by activating this collective vibration, these molecules, so to speak, swim and reach a diffusion coefficient which is 5 times the Brownian one, which is known in the field of active matter. And this could be interesting also, apart from the electrodynamic interactions, in a crowded environment, inside the cytosol, the excluded volume is very large. And this could be a helpful mechanism to accelerate diffusion in that case. Okay, let me stop here.